Hello everybody, welcome back to the lecture review series. Today we're going to be looking at lecture 9, which is talking about regulation and function of hemoglobin. This actually should be a relatively short lecture. There's a lot of information here, but I don't think all of it's very important. There's some aspects that are definitely important, but a lot of it is just very important at all. So yeah, we'll go over these things here, and I'll point out what's important, and hopefully it's helpful for your exam. So we've got the deoxy form of hemoglobin, which is the T state, and we've got the oxygen form of hemoglobin, which is the R state. So basically, when there's oxygen bound, it's in the R state. When it's not bound, it's in the T state. Right. So that's what you basically get from here. This is basically a review of last week's lecture, or not last week's, the last lecture that I went over. Right. Um, if you got a high P50, you got low affinity. If you got a low P50, you got high. Sorry, you got a high P50, you got low affinity. If you have a low P50, you have a high affinity. They actually edited this, which I actually didn't know. But there's something to be very aware of for hemoglobin is that because it's not it's because it's cooperative binding, the P50 value is actually also affected by this cooperativity number. So it says up here that KD only equals P50 when N equals 1. So for myoglobin, which only has one binding site, right, that's where that applies. But just be aware that they could sometimes change this, right? So if N is not just 1, it also changes for P50 as well. Um, yeah, I mean here we've got a range of PO2s, right? So you can see that from 0 to 8, right, is pretty much where we get like this maximum. Is a little bit more up here, but the slope from here to here is much steeper than here to here. So that's the benefit of cooperativity, right? Is that you can bind things very quickly and also release them very quickly. Yeah, so here's actually some really interesting Hill coefficient, right, is this N value, right? And so you can see here that as the number gets larger, the more sigmoidal the curve gets, right? And so as you get lower, like close to 0, right, the more like square this curve becomes, right? And so if the coefficient is you can imagine if it's negative, it's just gonna be like something like a square almost. It's gonna be weird. Like it's gonna be like this maybe, and like maybe like something like that, right? You can imagine that negative cooperativity looks far. Um, in this, I guess like allosteric, right? An allosteric binder, right, is something that binds to an allosteric site. So it's not the active site, it's not the main site of the protein where like the main ligand binds, but it's somewhere else on the protein. But that binding of this allosteric site affects the binding of the main site usually, right? Somehow, or it affects the protein in some way, right? So, like you said here, binding of one ligand at a binding site affects the binding properties of other binding sites, right? Modulates the activity of the protein, changes the shape, then changes the function, right? Allosteric protein, it's a protein with another conformation, right, induced by a post-translational modification. So, like, an example of this is O2 binding to hemoglobin favors the R state, right? So, this is just an example, right? So, O2 is the ligand, binds to this hemoglobin molecule, and it favors a certain conformation, the R state, right? If you remove the O2, it favors the T state, generally speaking, right? So, here we go. Just like I said, right? T state, lower affinity, R state, higher affinity, right? We have different types of allosteric effectors. There's homotropic and heterotropic, right? Homotropic is the same ligand, and heterotropic is different ligand, right? So, yeah, if, for example, if the binding of A affects B, right, that's a heterotropic effector. If the binding of A affects A, right, then it's a homotropic effector, right? Like in this case, right, hemoglobin, right, would technically be like a homotropic effector, right, because the binding of oxygen affects the binding of other oxygen, right. But something like a heterotropic would be like you'll see later is like a hydrogen ion. H plus binding affects O2 binding, which is a heterotropic because H plus is not O2, obviously, right. So if we look at the, we've got these different versions of hemoglobin, right? An interaction among the subunits in hemoglobin causes these conformational changes, right? And it alters the O2 affinity, right? And then, increasing or decreasing the O2 changes how hemoglobin responds to the oxygen, right? And so, like, because it's sigmoidal, right, it's able to release O2 relatively well and also bind it very strongly. This is an interesting slide. It kind of shows you the very minute difference. So you notice how like this is like a little bit more triangular shaped, right? This is a little bit more flat, I would say, right? The main idea here is that this is inside of myoglobin, right? So this is deoxymyoglobin. This is oxymyoglobin. O2 right here, right? It forms a sort of coordination complex with the iron. So in deoxymyoglobin, this histidine, right, has a coordination complex sharing its electrons with this iron, and it kind of pulls this heme group. This is the heme group here. It pulls the heme group upwards. However, when the O2 is present, it also shares its electrons with the iron and it pulls it back down. So it's almost like it's pulling it up and then this O2 is pulling it back down again, right? And the question is, what is the histidine connected to, right? So the histidine is connected to an alpha helix, right? And basically, right, what happens is when this O2 binds, right? So like normally under conditions, this histidine right here is pulling on this heme group, right? It's pulling it this way. But when this O2 binds, it pulls the whole thing this way, which also pulls the whole helix with it this way. And when you pull the helix, you enter the R state. If you get rid of the O2, right, this relaxes, right? If you get rid of the O2, like you get rid of this shifts back this way, and this also shifts back that way, and you enter the T state again, right? It's a very small change, but you think of it like a machine, right? If you change one thing, a lot of other things change. So this small change actually causes a lot of downstream effects in the protein that completely changes the structure of the protein. And the T state is actually stabilized, like otherwise it would never occur, right? By ionic interactions between subunits, right? So here's some examples, like there's an arginine, arginine here to aspartate here, there's an aspartate to an arginine. So the T state is stabilized by interactions, otherwise it would never occur in the first place, right? And this is the idea of cooperativity, right? So O2 binding to one subunit affects others, right? So adjustments in helix F, remember we talked about it, right? When O2 binds, it pulls that helix, right? And the binding O2 at the first site shifts the conformation of other subunits to the R state, right? So look at this, there's 4 different hemes, right? There's 1 heme, 2 hemes, 3 hemes, and 4 hemes, right? Keep that in mind, right? So the binding of 1, right? Pulling, like if 1, 2 bind O2, right? You pull one of these hemes, right? That small angstrom pull, right, actually completely changes the structure here, the structure here, the structure here, and ultimately results in the binding of more O2, right? You can see here, right? You can see like in the middle here, right, how the this histidine C3 here is there, but it's been shifted over here in the R state, right? So when you bind O2 at the first site, it causes these changes that ultimately affect all of these histidines, all of these sites, right? Yeah, I'm just trying to see if this is anything else I want to cover for this. First site shifts conformation of other subunits to R state. Yeah, I think that's the main thing I want to discuss here. Okay, this is where testable information really comes into play. This stuff is testable, obviously, but this is where it really becomes testable, right? So, H plus serves as a heterotropic effector, right? Talked about it before, right? H plus affects O2 binding. Not the same molecule, heterotropic, right? So, we know that your tissue makes CO2, right? And that combines with water to make bicarbonate H plus, which causes a pH decrease, more acidic, right? this pH decrease actually is able to bind to hemoglobin. So here we have the T state, right? That's not bound, right? And here we have the R state that is bound to O2, right? And the idea here is that when H plus binds, we actually promote the T state, right? So here we're in the R state, right? But adding H plus, right, actually decreases the affinity of hemoglobin for O2 because now we have something else that can bind to hemoglobin, right? So as H plus increases, the pH decreases, right? But this also means that the ratio of T to R state also increases. So there's more T state than R state, and affinity decreases, right? This is because this H plus basically, I don't want to say inhibits because it's not an enzyme. It basically competes with O2, but not. It's not like it's not a competitive inhibitor or anything like that. If you know what that is, but it's not competing with O2 for the same binding site, but it is preventing O2 from binding by inducing this T state, right? So the question is, how does it favor the T state? So here, right, one of the major contributions to this effect, which they call the Bohr effect, right, is the ionization of histidine at the C terminus of a beta chain, right? So in the T state, right, this histidine has a very high pKr, which means it's very basic. So now remember, histidine is usually around 6, right? But this is greater than 7, which is very, very high, right? pKr is very context dependent, right? It depends on what's around it, what amino acids are around it, right? Just like pKa, right? Or like acidity, right? You know, more electronegative things, right, will make things more acidic, right? And like more electron donating things will make things more basic, right? So there's probably a lot of electron donating groups around this histidine, right? Makes it more basic, right? Or something like that, right? So at relatively low pH, right, this histidine is positive, right? And is stabilized, right, by negative charges of aspartate, right? So these electron donating groups, remember, it's more basic, right? See how these aspartates, right, have electron groups on them that they can donate, right? Or they can use to stabilize histidine, right? So it stabilizes this base form, right? The fact that this positive histidine, right, is stabilized by these aspartates is what allows the T state, right, to be okay, right? So at low pH, right, with this extra H plus, the histidine is protonated. The aspartates nearby help stabilize this positive histidine, right, which leads to an increase in the release of O2, right? Under normal conditions, right, this histidine is never protonated, right? So this never happens in the first place. The other one we can look at is CO2, right? CO2 also serves as a effector, right? The interesting thing about CO2 is that it actually binds at the N terminus, right? So it's interesting, right? So it binds at the N terminus of these chains, right? Of these different subunits of the protein, right? And it releases an H plus, right? So the release of this H plus, right? Let's see if they talk about why. But the CO2 binding that releases an H plus, right, which also leads to the Bohr effect that we talked about up here, right, where this whole section here about the H plus binding to the histidine, right? There's that too, right? So obviously when tissue is using up O2, right, we end up having an increase in oxygen, a CO2 production, and if we end up producing a lot of CO2, right, it's gonna bind right to hemoglobin, right, and end up causing a release in O2, right, which is actually quite good, right, because you actually in the tissue, right, you want to release O2, right? So that's great. In the lungs, right, the O2 is high and the CO2 is low, right? So O2 ends up binding instead of CO2. But this is beneficial in the tissue, right, because there's a lot of CO2 and you want to squeeze every last bit of O2 out of the hemoglobin before you release it, right? Here's kind of like an example of how this looks, right? So notice, right, this is a nucleophilic attack, right? So if you remember any chemistry, right, there's 2 electrons here. These attack here, right? This comes up, right? And you have to remember that, now that you have that, right, you'll make an intermediate where this now has 4 bonds, right? And you need to release one of these H's, right? Which is where this H plus comes from, right? Which is how we get this Bohr effect, right? Oh yeah. And then this idea here that HB, hemoglobin, transports around 20% of the CO2 produced by the tissues, because obviously, because it's near the tissues, it'll absorb a lot of the CO2, right? The other thing to keep in mind is 2,3-bisphosphoglycerate, which is actually a glycolysis product. This also stabilizes the T state. There's one binding site for BPG per HB tetramer, right? So that's kind of something to be aware of, right? So the hemoglobin tetramer, right, has one binding site, right? And so basically, circulating. Let me actually double check something. I want to make sure, because like the idea, right? So because I want to make sure that that seems a bit odd. Yeah, there's one binding site. Okay, I just wanted to make sure, because this is kind of worded a bit weird. But yeah, basically each hemoglobin tetramer, right, has one is phosphoglycerate binding site, right, and this stabilizes the T state, right. And so basically, this is the structure of the 2,3-bisphosphoglycerate, which you will hopefully have a relatively good idea of what it looks like when you actually you guys start doing glycolysis, right? But yeah, so here's the idea, right? So BPG, right, serves as a high altitude adaptation, right? And you're probably wondering, what the heck? How does that make any sense? So let's think about it, right? If you BPG, right, stabilizes the T state, which means it would promote O2 release, right? So that would mean that you probably release more O2 to your tissue, and your tissue probably needs O2, right? So let's go ahead and take a look at if that holds true, right? So at high altitudes, right, it makes sense. You can't just magically produce more erythrocytes, right? It takes a while, right? You can't just increase hemoglobin content per cell. Also takes some time, right? This is the fastest response, right? Because glycolysis is happening, and you're gonna produce 2,3-bisphosphoglycerate, right? As a result of the 1,3-bisphosphoglycerate that's actually in the glycolytic pathway being turned into 2,3-bisphosphoglycerate through a mutase of some kind, right? Which you guys will learn about later on. So BPG, right, is negatively charged, as shown in the structure, right? 1, 2, 5, right? Very negatively charged, right? And it binds, right, to the positively charged site in the T state, right? This site is not there in the R state, so it cannot bind in the R state, right? But the BPG binds to this site in the T state and keeps it stuck in that T state, right? The positively charged groups, right? And BPG gets stuck there in the middle, right? In the R state, see this, that space is not there, right? But BPG basically wedges itself in here and stops it, right? That's how BPG kind of changes, it induces the T state, right? See here, here's some examples of how this works here. So like you can see here at sea level, right? If you have more BPG, 8 millimolar BPG versus 5 millimolar, right? See how the curve shifted very slightly, right? Right. So the affinity is a little bit lower when you have more BPG than if you have less BPG, right? And the interesting thing is, like, if you were to take 2 points, right? Let's say you're at sea level, right? For both of these, right? You're about max, right? And this is on your lungs, right? In the tissue, you're about here. Notice, right, that the drop from here to here is probably a little bit more than it is from here to here, right? So notice how we're actually technically releasing a little bit more O2, right? When we shifted this, right? Because we have shifted to the right, we're actually binding about the same amount of O2 at sea level, right? But we're actually releasing quite a little bit more, right? Same thing here, right? We're binding about the same, probably a little bit less, right? But releasing around the same, maybe a little bit more, right? That's why it's a good adaptation, right, at higher altitudes, because we're able to bind that oxygen, right, and then release it very easily. Carbon monoxide. So carbon monoxide is interesting. It actually technically increases infinity, but we'll get to that in a second, right? So carbon monoxide binds in the same position as O2 very strongly, right? 250-fold higher. My God, that's a lot, okay? So the PCO in the air, though, is very low. There's, it's not a lot, right? But for smokers, there's actually 1% of their hemoglobin sites are actually filled up with carbon monoxide. But yeah, eventually, carbon monoxide does build up over time, right? And it is fatal at 60% occupancy. So, if you have 4 sites, right? If you have like 2 of the sites filled up with carbon monoxide, it can be fatal, right? A little bit more than 2 sites, right? So because it has such a high affinity, it doesn't dissociate, right? So it actually lowers the number of O2 binding sites available, right? But what's interesting? What's interesting about this, right, is that this whole hemoglobin mechanism with the iron, right? O2 also does that, or carbon monoxide also does that, right? So it actually does increase the binding of O2, right? But it's so tightly bound on there that it doesn't let any O2 replace it. So it increases the binding of O2 to other spots, but it stops O2 from binding to the spot that it is now sitting in, right? If you look here, right, CO binding is allosteric, affects O2 binding at other sites, right? So it binds to a spot where O2 is supposed to bind and then affects O2 binding at other sites, right? So does it bind with higher or lower affinity? The answer is higher. So if you look here at this CO version, you can see, right, that the P50 is actually around here, while up for these, it's actually around here, right? However, what you should notice is because we are taking up sites, right, 50% of carbon monoxide, because we are taking up sites of where O2 can bind, we're actually dropping the possible max fractional occupancy available to us. So although the affinity is slightly higher, right, if you have carbon monoxide, it's actually detrimental because you're actually just binding less O2 in general, right? So that's why it's deadly, even though it does seem to cause a slightly increased affinity, right? Seems like it increases the affinity, right? And it does increase the affinity, right? But it doesn't matter if you increase the affinity if you're dropping the amount of O2 you can hold in the first place, right? We need oxygen to survive, and if we're dropping the amount of oxygen we can actually carry in our hemoglobin, we have to breathe more and more, right, to get more oxygen, right? And we basically are probably gonna start hyperventilating, right? Try and get that oxygen that we need, right? So here's just a summary of how things work. You got the T state, low affinity, R state, high affinity, right? And this kind of T-R state ratio changes with response to physiological changes. This is what I would say you should keep in mind. This is a very nice slide. It really puts together what you should think about. So, like for O2, for example, right, it shifts to the R state, right? And it makes it so that the affinity for oxygen the P50 decreases, right? Which means that the concentration required to have O2 reach 50% occupancy decreases. So we have a very high affinity, right? So less O2 required to reach 50% occupancy. H plus, it is a heterotropic effector, induces the T state, increases P50. We need more oxygen to actually get to 50% occupancy. Same with CO2, right? Binds to the N terminus, right? This H plus binds to the histidine, right? This CO2 binds to the N terminus, causes a Bohr effect that releases H plus, so kind of a double whammy, right? Induces the T state, right? And increases the oxygen required to reach 50% occupancy. 2,3 BPG, right? Stabilizes the T state, has a bunch of negative charges, and interacts with the positive charges on the hemoglobin, right? Which results in the stabilization of the T state, which increases the P50, right? Same thing as we said before. You need more oxygen to reach 50% occupancy. And this one is the weird one. Carbon monoxide, right? Carbon monoxide actually stabilizes the R state. So it actually makes it more likely to bind oxygen, but and it actually decreases the affinity, or sorry, increases the affinity. I keep messing that up. Increases the affinity for hemoglobin, right? It decreases the P50, meaning we need less oxygen to actually bind to, less oxygen required to actually bind to hemoglobin, right? To reach, I guess, less oxygen to bind to, my gosh, less oxygen to reach 50% max occupancy, right? However, what this image doesn't tell you is that carbon monoxide actually decreases the max O2 you can actually hold, right? So if you have 50% carbon monoxide, right, instead of having 4 binding sites for oxygen, 2 of them have now been filled with carbon monoxide, and only 2 more are left, right, to actually bind the oxygen that you need to breathe, right? So the issue here isn't about the affinity and which state it's in. It's actually about how much oxygen you can bind, period, in the first place, right? Because if we've gone from, even though you're in the R state, because carbon monoxide binds so strongly to hemoglobin at 250 times the affinity, right, 250 times the strength of oxygen, it doesn't matter if you have a higher affinity for oxygen now. Over time, you will decrease the number of available spots open for oxygen to bind at all, and therefore you'll go from carrying 4 oxygen molecules to 3 to 2 and then to 1 until you're at none for that hemoglobin, right? That is the issue with carbon monoxide. Other than that, there isn't really much else after this lecture. It's a relatively short lecture. Hopefully it was helpful. I think this is one of some of the more testable content, so I would keep this in mind. Very possible that you could be tested on these, and actually I'd be very surprised if they didn't test on these, right? This is very like easy stuff to test on, right? You can make like multiple choice questions, like some very simple like I guess like does this stabilize the T state or does it stabilize the R state, right? If you look at my exam review, there was a question about, I think, hydrogen ions. Does it stabilize the T state or the R state, and how does that affect like O2 dissociation, right? So just make sure you're studying these and keeping these in mind. But yeah, hopefully this video was helpful, and I'll see you in the next one. Happy studying.